In this video, we are going to study about the structure of proteins. We know that proteins are the polymers of alpha amino acids. So, hydrolysis of protein will give alpha amino acids. Now, these alpha amino acids are linked by a bond or by a linkage called peptide linkage or peptide bond. So, let us see what you mean by a peptide bond or peptide linkage. So, what do you find this peptide bond or peptide linkage? Between the amino acids in a protein molecule. Now, what do you mean by a peptide linkage or a peptide bond? Peptide linkage is an amide. And you know amide is CONH. So, peptide linkage is an amide formed between carboxylic acid of one amino acid molecule and amino group of another amino acid molecule by loss of a water molecule. So, we get a peptide bond or a peptide linkage. How is that formed between two amino acids? By the loss of a water molecule, and the water molecule is lost from carboxyl group of one amino acid molecule and amino group of another amino acid molecule. So, let us see how a peptide bond, that is CONH bond, is formed between glycine and alanine. Now, glycine, I am going to draw the structure of glycine. Glycine is NH2, CH, COOH, and we have R here, and you know the R in glycine is hydrogen. So, I am going to replace that as CH2. So, this is glycine plus alanine. Alanine we have NH2, CH, COOH, and the R in alanine is methyl group CH3. So, this is alanine. Now, these two, when they combine, H2O is removed, minus H2O. Now, from where is H2O removed? The OH group of COOH and hydrogen, one hydrogen of alanine. They will be removed as H2O, and the product formed is NH2, CH2, CONH, CH, COOH, CH3. And you can see this is our peptide bond. Okay, and you can name this product as, here glycine and alanine combined. So, the product is known as glycyl alanine, or by using a three-letter word you can write as glycyl alanine. Now, here two amino acids combine to give how many peptide bond? One peptide bond. So, we can call this as a dipeptide. Dipeptide. Now, when I say dipeptide, how many amino acids combined? Two amino acids combined. And how many peptide bond is present? One peptide bond. So, a dipeptide is a peptide bond, or it is formed when one or two amino acids combine, we get a dipeptide. And here the name of the dipeptide formed is glycyl alanine. Now, we can have tripeptide, tetrapeptide, then pentapeptide, hexapeptide, etc. When I say tripeptide, we have three amino acid molecules that combine to form a peptide bond. And how many peptide bonds are formed? Two peptide bonds. Now, tetrapeptide means four amino acids has combined, and how many bonds between four amino acids, peptide bonds? There are only three peptide bonds. So, we can have hexapeptide, pentapeptide, etc. Now, when a number of such amino acids is more than 10, then the product is called a polypeptide. So, if the number of amino acids is more than 10, greater than 10, we can call it as Polypeptide. Polypeptide. Now, again we are going to see a definition for protein. Already we discussed proteins are the polymers of alpha amino acids. Now, next definition is a polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 U is called a protein. So, if we have more than 100 amino acids, we can call it a protein. So, what do you mean by a protein? A polypeptide with more than 100 amino acid residues having molecular mass higher than 10,000 U. U means unified mass. It can be called a protein. Okay, so this is thus we come to know that these proteins are made up of amino acids, and the amino acids are linked to each other by a peptide bond or peptide linkage. Now next let us see the classification of proteins. Classification. Now proteins can be classified into two types. The first is fibrous protein and the second type is globular protein. Now, what do you mean by fibrous and globular protein? Now, fibrous protein means when a polypeptide chain run parallel and are held together by hydrogen and disulfide bond. We call it a fibrous protein. So, in fibrous protein, we can say that the polypeptide chains run parallel and are held by hydrogen bond and disulfide bonds. Now, they have fiber-like structure. So, that is the next point coming under fibrous protein, that is fiber-like structure. Now, the next point is these fibrous proteins are insoluble in water. And last is example for fibrous protein, that is keratin. Keratin is present in hair, nail, wool, silk, etc. And next example is myosin, present in muscles. So, these are the two examples for fibrous protein. Now, next let's see what are globular proteins. In globular proteins, the polypeptide chains coil around. So, here the polypeptide chains coil and the shape is spherical, and the globular proteins are soluble in water. And an example for globular protein, there are two examples. One is insulin and the second example is albumin. So, these are the two examples for globular proteins. Okay, so these are the differences between fibrous and globular protein. So, fibrous protein has polypeptide chains run parallel and they are held together by hydrogen bond and disulfide bond. Globular proteins, the polypeptide chain coil around. Then fibrous protein has fiber-like structure and globular protein has spherical structure. Fibrous proteins are insoluble in water and globular proteins are soluble. Example for fibrous protein is keratin, myosin, and globular protein is insulin, albumin. Now next let us see the structure of proteins. So next we have the structure of proteins. Structure of proteins. Now the structure of proteins can be studied at 4 different levels. So we have primary structure, secondary structure, tertiary structure and quaternary structure of protein. So let's study first the primary structure of protein. Primary structure of protein. Now in primary structure, the polypeptides in protein has amino acids linked with each other in a specific sequence. Means there is a particular arrangement of polypeptide chains in primary protein. And that is said to be the primary structure of protein. So there is a particular arrangement, or we can say the amino acids amino acids are linked in a specific sequence, and if there is a change in any of these arrangement or sequence, then it will cause change in the primary structure. Okay, which means it will give rise to a different protein. So that is the primary structure. So in primary structure, the amino acids are arranged in a particular sequence. Now next comes the secondary structure of proteins. So second is secondary structure. Now secondary structure of proteins is found to exist in two types. One is alpha helix and second is beta plated sheet structure. So secondary structure is of two types. One is alpha helix and second is beta plated sheet structure. So let's see what is alpha helix and beta plated sheet structure. Now alpha helix structure, it arises, or both these structures, alpha helix as well as beta plated structure, both arise due to regular folding of polypeptide chain due to hydrogen bonding between CO and NH groups of the peptide bond. So both these structures are obtained by folding of the polypeptide chain, and when you fold the polypeptide chain, a bond is formed between the CO group and NH group, and that bond formed is hydrogen bond. Now how is the alpha helix structure formed? I told you both are formed by the folding of the polypeptide chain. Now alpha helix means it is also formed by the folding of the polypeptide chain, and in case of alpha helix structure we get the maximum number of hydrogen bonds. So in alpha helix structure, maximum number of hydrogen bonds are obtained by twisting that polypeptide chain into a helix, so that NH group of each amino acid residue is hydrogen bonded to CO group of an adjacent turn of the helix. So when you draw the alpha helix structure, we can draw like this. Polypeptide chains is folded, and here the CO group in one helix will form a hydrogen bond with NH of another helix. So this is alpha helix structure. So in alpha helix structure we have the maximum possible number of hydrogen bonds. Now next we have the beta plated sheet structure. Now in beta plated sheet structure, the polypeptide chains are stretched out to maximum extent, which are laid side by side, and they are held together by intermolecular hydrogen bonding. So in beta plated structure, their polypeptide chains are stretched out to a maximum extent, so that a hydrogen bond exists between CO group and NH group. Now these structures resembles the plated folds of drapery, and so it's known as beta plated structure. So this is the secondary structure of protein, that is alpha helix and beta plated structure. Both are formed by the folding of the polypeptide chain, and alpha helix structure when folded give rise to maximum number of hydrogen bond between CO and NH group. Now next we have the tertiary structure of protein. So C is tertiary structure. Now tertiary structure is obtained by the folding of secondary structure. So it is more complicated, the tertiary structure. Now that further folding of secondary structure give rise to tertiary structure, and here in the tertiary structure we get two major proteins, that is the fibrous and globular proteins. Now the main forces which stabilize the secondary and tertiary structures of proteins are hydrogen bond, disulfide linkage, van der Waals force, and electrostatic force of attraction. So this is a board question. Which are the forces that stabilize secondary and tertiary structure of proteins? They are hydrogen bond, disulfide bond, van der Waals force, and electrostatic force of attraction. Now the last structure is quaternary structure of protein. Quaternary structure of protein. So quaternary structure of protein is obtained by further folding of tertiary structure. We get a quaternary structure, and the quaternary structure is highly complicated. So these are the 4 levels at which the structure of protein is studied. Now next we have a term called denaturation of proteins. So next we have denaturation of proteins. Now, before we come to the word denaturation of proteins, let us see what you mean by a native protein. Native protein means proteins found in biological system with a unique 3-dimensional structure and biological activity is called a native protein. Now, what do you mean by denaturation of protein? When a protein in its native form is subjected to physical change like change in temperature or chemical change like change in pH, the hydrogen bonds are disturbed. Due to this, the globules unfold and helix get uncoiled and protein loses its biological activity. This is called denaturation of protein. So, when protein is subjected to a physical or chemical change, what will happen? The globules unfold and the helix get uncoiled and protein loses its activity. This is called denaturation of protein. And as a result of denaturation, the secondary and tertiary structures are destroyed, but primary structure will remain intact. There is no change for the primary structure, but secondary and tertiary structure of protein will get destroyed. And an example, there are 2 examples for denaturation of protein. One is coagulation of egg white on boiling. Coagulation of egg white on boiling is an example. Also another example is curdling of milk, which is caused with the formation of lactic acid by the bacteria present in milk. So these are 2 examples for denaturation of protein. Now we have 2 questions to discuss. The first question is, where does the water present in the egg go after boiling the egg? Now the answer is, we know that water present in the egg is the protein part. Now the egg protein will first undergo denaturation and then coagulation. When the egg is boiled, the water present in the egg gets absorbed and adsorbed in the coagulated protein through hydrogen bonds. Now the next question is, the melting point and solubility in water of amino acids are higher than that of corresponding halo acids. Explain. Halo acids means carboxylic acids containing halogen atom. So the melting point and solubility of amino acids in water are higher than halo acids. Now the answer is, amino acids have strong electrostatic attraction or strong dipole-dipole attraction because of their dipolar salt-like character. We know amino acids, they exist as switterion, dipolar ion. They interact strongly with water due to their salt-like character, and they can form intermolecular hydrogen bonds. Okay, so due to that switterionic property of amino acids, they are more soluble in water than halo acids. So this is about the amino acid peptide bond, then classification of proteins and structure of proteins and denaturation of proteins. So with this we complete the topic, that is proteins. Thank you.